Isoprene is a volatile compound produced in vast quantities by many organisms, including to a small extent humans, but especially by plants. In fact, as much isoprene is liberated into the atmosphere every year as the greenhouse gas methane. Isoprene is a Jekyll and Hyde gas. On the positive side, it encourages cloud formation that counters global warming. But on the negative side, it's so reactive that it mops up many of the chemicals that would otherwise react with methane, and so it exacerbates global warming. Also, it encourages the production of ozone, which can damage human and animal health. Unfortunately, the big producers of isoprene include tree crops that are being grown throughout the world for biofuel production, especially in the tropics, oil palms, and in temperate regions, willow and poplar trees. We know very little about the microbes that use isoprene as a source of food. We're driven to understand who consumes isoprene, how they do it, and whether we can harness this activity to control the release of isoprene to the atmosphere.
We are trying to determine how much isoprene is degraded by microbes. We are doing this by measuring the degradation rate in soils and on leaves. Soils are expected to remove about 5% of the isoprene produced. Leaves are the primary source of isoprene production, and therefore we expect them to host isoprene degrading bacteria. To test the degradation rate, we spike soils and leaves with low concentrations of isoprene and an inert tracer. We sample the gas over time, concentrate it using a liquid nitrogen cryotrap, and analyse it using gas chromatography. This separates out the compounds and can tell us how much isoprene there is left. Through this, we should be able to build a better picture of the isoprene cycle, and we have already shown that isoprene can be degraded within minutes in soils.
One of the main goals of the Isoprene group is to find out exactly how isoprene is metabolized in the environment. To do this, we use cultivation-dependent methods to try and isolate those key organisms that play a major role in these environments. For example, from soil samples and from leaf washings from isoprene-emitting and non-emitting trees. Most organisms cannot be grown in the lab using the current conditions. So for this, we change the conditions a little bit, for example, changing the concentrations of the media, changing the temperature we grow them in, to try and isolate these organisms best we can.
As mentioned before, not all organisms can be cultivated. Therefore, to get a better picture of the organisms in our samples, we have used cultivation-independent techniques such as stable isotope probing, next-generation sequencing, metagenomics, and metatranscriptomics.
We've now got several isoprene degrading strains growing in the laboratory, and we're using a variety of molecular methods to identify the isoprene degrading genes that are involved. Firstly, we sequence the genome of several of these bugs, and then by studying the genome sequence, we can identify which genes are likely to be involved in isoprene metabolism. And we can verify this by a suite of other molecular methods, including transcriptomics and proteomics. Then this sequence information feeds back into our environmental work, and we can identify the genes that we can then retrieve from the environment and use in our cultivation-independent methods to identify isoprene degraders.
So using our cultivation-dependent methods and our cultivation-independent techniques, we're actually building up a picture of where these organisms are in the environment and the processes that they carry out in the environment with regards to isoprene. So we've got a whole collection of isoprene-degrading bacteria which we're starting to study. We're looking at the physiology and biochemistry of these organisms. We're also looking at the isoprene degradation pathways, in particular in one key organism which is called Rhodococcus. And again, the picture that's emerging is that the Rhodococcus can actually degrade the isoprene quite readily in the environment. So we know quite a lot now about this key organism. What we want to do is to find out more about organisms present on leaves and present in the soil and how they contribute to the global isoprene biogeochemical cycle. And in this way then, we may well be able to predict and even mitigate the effects of isoprene release into the atmosphere.